All right, students, so I figured that I would make a video that explains to you exactly how the test is going to go down for our class. So what I'm going to do is I'm going to put here a strand of DNA. This is how small of a strand of DNA you will be working with. And we're going to go through these steps to make sure that you can be successful. And we're also going to make reference here to our rubric. Of course, as I mentioned before, you are allowed to use your slides, and you can use anything inside of your notebook. I'm going to say that in particular, pages 11 and 12, where you have your images of transcription and translation, are going to be the most helpful for you, as well as your slides. And we're going to use these to show that you can get an 100%, a perfect score on this test, using only these resources and the skills that you have learned along the way. Okay, so let's start here with step 1. Okay, so here on the rubric, it says, right, the first thing that you're being asked for here is to say, show where transcription takes place in a cell. So if we look here at our slides, on the first slide here, it says transcription is in the nucleus. You can also see here in our notebook, right, that we are looking here at the nucleus. We're coming in closer to the nucleus. So we are in the nucleus for transcription. That is where transcription takes place. So if you told me that transcription takes place in the nucleus, you would receive the point here for this rubric. So check here. The second thing that's going to happen is I'm going to give you a strand of DNA, and I'm going to give you just one of them, and I'm going to ask you to write the complementary nucleotides for it. Based on the slides that you have here, you should be able to resolve this question. So I might give you something like this. So here we have a strand of DNA. So I'm going to ask you to write, as it says here, write the complementary strand of DNA. So as we know from our images inside of our notebook, and also from here, right, we know that in DNA, adenine forms base pairs with thymine and cytosine forms base pairs with guanine. We also know here from our images, right, we have our images here where we see that C and G pair and A and T pair. So wherever there's a C, I'm going to put a G. Wherever there's a G, I'm going to put a C. Wherever there's an A, I'm going to put a T. And wherever there's a T, I'm going to put an A. So I'm going to use these base rules here to help me. So for every A, I'm going to put a T. For every T, I'm going to put an A. A goes with T. T goes with A. G goes with C. G goes with C. G goes with C. G goes with C. T goes with A. C goes with G. T goes with A. T goes with A. A goes with T, A goes with T. So I've written the complementary strand of DNA that would earn me the next point on our rubric. So now we want to show two base pairs of DNA. So again, base pairs are just the combination of those two nucleotides. When they bind together, you have this image inside of your notebook, right? The base pairs are A and T and C and G. So all I have to do is put a square around those and I can get my points. So I have my A and T, or T and A. Here's one base pair, and here's another base pair. So if I can do that, I will receive the next point on the rubric. Next, we want to show what RNA polymerase does to DNA. So if we look both at the images inside of our notebook, right, we see that the RNA polymerase, what it did was it wrapped around the DNA and it separated the two strands of DNA to add in an mRNA. We also see this in our slides. Step 2, RNA polymerase wraps around and separates the two strands of DNA. So we're going to show that here too, right? Just like we did before, here is this template strand. So what I'm going to do is I'm going to put this RNA polymerase right around this DNA molecule. I have now separated the two strands of DNA. Great. And so if I can do that, I will receive the next point, and I will receive all the points for this portion of transcription DNA. I'll receive 4 points. Great. Next, it's now asking, so we're going to now see what happens. What happens when we now make our mRNA? That's the next step. Right? Step 3 of transcription is RNA polymerase adds a complementary nucleotides to form an mRNA, and these are the base pairing rules. So let me slide in this mRNA here, as we have done many times before, and let's write in our base pairs. So in mRNA, we have a little bit different. So on our DNA, if we have a letter T, we're going to add the letter A on RNA. If on DNA we have a G, we're going to write the letter C. If on DNA we have a C, we're going to write a G. And if on DNA we have an A, we're going to write a U. So this is the key difference here. So since there is a T here, I'm going to write that this goes with A here goes with U, T goes with A goes with U, C goes with G, C goes with G, C goes with G, C goes with G, A goes with U, G goes with C, A goes with U, A goes with U, T goes with A, T goes with A. And we now have our strand of mRNA. So, right, we followed the base pairing rules from here. So we can go ahead and show that we get our checkpoint here. We have the complementary nucleotides for RNA. So now it's going to say, discuss how RNA base pairs are different from DNA. So in DNA, as we saw before, right, on DNA, the base pairs that we have are that T and A base pair, as well as G and C. These base pair with each other. Thymine base pairs with adenine, guanine base pairs with cytosine. mRNA, on the other hand, instead what it does is, when there's a thymine on, right, as we said before, when there's a thymine on DNA, we get an adenine on RNA. When there's an adenine on DNA, we get a uracil on RNA. When we have a thymine on DNA, we get an adenine on RNA. And when we get a cytosine on DNA, we get a guanine on RNA. And when we get a guanine on RNA, guanine on DNA, we get a cytosine on RNA. So these are the same base pair rules that we have here. And that's the difference, right? So DNA and RNA are different because DNA, if you look at these right next to each other as well, right? These are, this strand up here and this strand here are identical, except for instead of a T, you have a U. So in all these places where it's identical, otherwise, the only difference is that instead of T's, we have U's, uracils. Great. So we would get this point for being able to explain that. Then it says, discuss how the number of strands from RNA are different from DNA. So RNA here, we have only one strand, whereas DNA here has two strands. Right? This is a key difference between the two pieces. So DNA has two strands, RNA has one strand. And that would earn me this point. The result of transcription, right? So at the end of transcription, what we have made is we have made an mRNA. This is the key end piece of this process. We have finished transcription with step 4 here, and we have made our mRNA. And we now move over to translation. So the key product, the key result of transcription is an mRNA molecule. And that would earn me that point. I would earn 4 points here. Translation part 1. All right, so now we have our mRNA, right? We have our DNA that stays in the nucleus and our mRNA that has just been made. I seem to have misplaced my polymerase, but the polymerase would also be here in the nucleus as well. It's not too important for what we're doing here, but that is where it would be. So in translation now, what we're going to do is we're going to look here at our slides, and we see that in translation, which by the way happens in the cytoplasm, this here asks you where translation takes place in the cell, the mRNA leaves the nucleus and enters the cytoplasm. Here it will find an organelle called a ribosome. So this mRNA, which has been in the nucleus this whole time, now goes over to the cytoplasm. And here is where the translation of this mRNA is going to happen. So if you were able to say that translation happens in the cytoplasm, you would have received this first point. Okay, then it says to discuss the role of the ribosome. So here on the second slide of translation, it says the ribosome reads the mRNA strand until it finds the nucleotide sequence AUG. This is called a start codon. So I'm going to take here my ribosome and I'm going to take my mRNA, and my ribosome is going to read this mRNA strand, and I'm going to read it until I get to AUG. This is called a start codon. So I'm going to circle AUG here on my mRNA strand inside of my ribosome. Okay, so that's what it was. And I also have identified the start codon being AUG. AUG is always going to be the start codon. Another way for me to do that for myself is to show this here, right? So in this table that we've looked on a bit, right? Start here, start codon is this M, the start amino acid is an M, and it's right next to the codon AUG, which is also right here, AUG. So we have discussed the role of the ribosome in reading the mRNA strand and in identifying the start codon. Okay, the next step of this is now we want to be able to identify the anticodon that is going to go with the start codon. So again, we can look here at our slides, but we can also just use the fact that we know about base pairing rules of RNA to help us in this spot. So what I'm going to do in this part of the test is I'm going to give you a blank tRNA molecule with a blank anticodon. So you're going to write the correct sequence for the anticodon. And to do that, right, we have this here. You can find this here. So it says a tRNA molecule with an anticodon with nucleotides UAC connects to the codon. It carries the amino acid called methionine, MET, or M. So as this actually just gave us the answer, right? It says that the nucleotides are UAC. So we're going to write here just UAC. UAC. Right? A goes with U. Let's see, UAC. So A goes with U. Or U goes with A goes with U, C goes with G. Great. And it's going to carry methionine. And again, we can also look here at our table, right? AUG is our codon. AUG will give us the letter M, and M is methionine or MET. So I'm going to grab my methionine, and I'm going to place it on my tRNA. Here at the top of my tRNA. The T in tRNA stands for transfer, so it's going to transfer this amino acid over to this strand here, which is going to be a peptide or polypeptide. It's eventually going to be a protein, but we're getting ahead of ourselves. So let's go to the next three letters. The next codon is GGG. So let's find what the anticodon would be. So as we know from our base pairing rules, right, with RNA, G goes with C, G goes with C, G goes with C. You can also, again, use the images inside of your notebook that would show you this as well. So in our notebook, right, when we had these images, we showed that, right, our G goes with C, G goes with C, U goes with A, etc. Right? We have this information here as well to help guide us. But our anticodon is CCC. Our codon is GGG. So let's look here at our amino acid table to find out what amino acid that would be. So if we have our codon being GGG, here we look at first letter G, second letter G, third letter G. GGG is for the letter G. G is glycine or gly. So I'm going to take a glycine and I'm going to put it right here on top of my tRNA molecule. My tRNA is going to transfer this amino acid of glycine over to my peptide. So this is now called a peptide. Again, if we looked here at our notebooks, that can help us as well. If we see on page 12, a peptide is when we have a sequence of amino acids. It's also called a polypeptide. It's the same thing, really. But you can also look here on step 4 of your slides, where it says the tRNA molecule adds the methionine amino acid to form a chain called a peptide. And this is what it's called. So a peptide is a chain of amino acids, and it's kind of the intermediate step between a amino acid and a protein. All right, but let's continue going. So let's move this guy out of the way and go to the next codon. We have UCU. So I'm going to circle UCU. This is my next codon. And I'm going to write the anticodon. So U goes with A, C goes with G, and U goes with A. And here on this tRNA, this UCU, well, the first letter being U, second letter being C, third letter being U, it means we have the letter S. S here is for serine or ser. So I'm going to grab a serine amino acid, and I'm going to place it here on top of this tRNA molecule. Again, the T in tRNA stands for transfer. I'm going to transfer this serine over to my polypeptide or my peptide. I'm going to go on over to the next 3 letters, the next codon, which is UAA. UAA, if I look here, if I write my anticodon on my tRNA molecule, so the letter U goes with the letter A goes with the letter U, and the letter A goes with the letter U. Then I'm going to go here to my chart and find AUU. First letter A, second letter U, third letter U gives me I. I is isoleucine or ILE. And so, oh shoot, I made a mistake. Look at that. So we should be looking here for our codon, not our anticodon. Good catch, Prof. Alexis. So here we're going to find that, right, UAA first letter U, second letter A, third letter A is stop. That's a stop codon that tells us to stop this process. Sometimes you can put a little stop sign there. I have a stop sign that you can use if that is helpful for you to use. You don't have to, but you can put a stop here to stop this process. Okay, so let's go back here, right? So let's just make sure that we showed that we have the anticodon that goes with the start codon, right? Our start codon was AUG, and the anticodon that goes along with it is UAC. So we were able to successfully identify our anticodon. And you were also able to do that, right? Just by looking inside of your notebook on the images that you colored in, right? AUG went with UAC, and UAC is what we have here too. They match up. Okay. So let's now say, so it says you can successfully use and identify the vocabulary tRNA, amino acid, peptide, and protein. So again, by looking at the images inside of our notebooks, we can see that what a tRNA molecule is it's this thing that kind of looks like a fork. These are called tRNAs, transfer RNAs. What they do is they transfer information from the RNA over to make a protein. So this here, this fork-looking thing here, is a tRNA. Okay, then the next one, it wants it, so we were successfully able to use tRNA, great. tRNA has it carries an anticodon, and it also carries an amino acid over to a peptide chain or a protein. Okay, next, so amino acids are these guys here. Amino acids are little molecules that are carried by tRNA molecules that will eventually make a protein. So these methionine, glycine, and serine here, also, in case you were wondering, these are all the amino acids that we need in our body. Those are all amino acids, and those are carried by our tRNA molecules. So we were able to use tRNA, we will use amino acid correctly. So we'll get our point for amino acid. Peptide, so as we showed before, peptide is just when you have a chain of amino acids. You can also find it here, right? So in step 5 of our process, of our procedure here for translation, it says, the process continues until the ribosome finds a stop anticodon or stop codon. The chain of amino acids, also called a peptide, this is a peptide, it's a chain of amino acids, is released and forms a protein. So a protein is the final product. It is the translation of an mRNA into the amino acids that correspond to that mRNA. So we were able to use both the words peptide and protein. And we got 4 points for both of these sections. So we got a 4 on our test here. As I said before, you do not have to take this test in English if you feel unprepared for doing so. But if you do it in English, you get an extra point. So what you would do is instead of getting a 4, you would get an extra point. So I would give you a 5 out of 4, which roughly translates to about like 120%. Anywho, hopefully this helps you a bit. We will see you tomorrow.